Hey guys, welcome to my YouTube channel Knowledge of Biology with Anjali Bute. Today I am provide you notes about detection of molecule using RIA. RIA means our radioimmunoassay. This technique is used to detect and measurement of peptide, vitamins, for example insulin. Okay, so look at this diagram. In this technique, firstly we are take by antibodies. For example, these are our antibodies and this is our antibody binding site. Also take by radio labeled antigens. They are mixed to each other properly in this solution. Okay. Then we are add unlabeled antigens from patient blood samples. Okay. After few time we are observe it unlabeled antigens are carry binding with our antibody than labeled antigens. Okay. And this solvent are automatically change their color because carry binding, competitive binding in this solution. So unlabeled antigens are separated. Okay, radioactive antigen bound with antibodies are measured with help of gamma rays in this technique. Okay, so observe there. First point, radioimmunoassay RIA is highly sensitive in vitro technique used to detect and quantified molecules, typically antigen, hormones, drug or proteins. Means in this technique we are used to separate it mostly in hormones, drugs, proteins. Okay, by measuring radioactively labeled tracer molecule in a competitive binding reaction. Look at this solution. Carry to competitive binding means unlabeled antigens and labeled antigens with antibodies, with specific antibodies. Then second point is the amount of labeled antigen bound is inversely proportional to the amount of unlabeled antigen in the sample. Mostly unlabeled antigens are carry binding with our antibodies. Competitive binding between known amount of radio labeled antigen and unknown amount of unlabeled antigens means we are not known to be unlabeled antigens and exactly their components, but they are carry binding to our antibodies. Okay, the analyte for a limited number of antibody binding sites, so we are easily detected by our samples, for example hormone, insulin, etc. The release on the principle of competitive binding, where unlabeled antigen from a sample compete with a fixed amount of labeled antigen for a limited number of antibody binding sites. Then a mixture of sample containing unknown unlabeled antigen of a known amount of the radioactive labeled antigen and a specific antibody is incubated. Binding competition in the unlabeled antigen. Unlabeled antigens also called as a cold antigen for a sample compete with the labeled antigen, hot antigen for a binding to the antibody. Really are carried to they are the competition between to be each other unlabeled and labeled antigens for the binding of the antibodies. Next point is amount of radioactive is inversely proportional to the concentration of labeled antigen in the sample. Higher sample concentration leads to less radioactively binding. Then it is a highly sensitivity techniques. Okay, totally depending upon higher concentration about the competitive binding between the labeled and unlabeled antigens to the antibody. Then last point is RIA is used to measure hormone, for example insulin, vitamin, drug and tumor marker. That is a very beneficial technique to detect the diagnostic. Okay, for mostly in tumors. It is the also crucial for the diagnosis allergy, specifically uh, detection and autoimmune disease also. So that is our main and important points about the radioimmunoassay. I hope you understand these diagrams, and these are very important notes about the detection of molecule using RIA. Thank you so much.